Imagine standing inside a modern pharmaceutical research laboratory. Around you, automated instruments are screening thousands of chemical compounds against biological targets. Genomic sequencing machines are generating vast streams of genetic information. Clinical trial platforms are recording patient outcomes, laboratory measurements, imaging results, and biomarker data from hospitals around the world. Every day, biomedical science produces an extraordinary volume of information. Hidden inside these datasets are patterns that could reveal how diseases work, how drugs interact with biological systems, and which therapies have the greatest potential to improve patient health. But there is a challenge. The scale and complexity of biomedical data have grown far beyond what traditional analysis methods can easily interpret. A single drug discovery campaign may involve millions of molecular measurements. A clinical trial may include thousands of patients, each described by hundreds of biological variables. Within this immense ocean of data, identifying meaningful patterns becomes an extraordinary scientific challenge. This is where machine learning enters the story. Machine learning is a branch of artificial intelligence that enables computers to learn patterns directly from data. Instead of relying on explicitly programmed rules, machine learning systems analyze large data sets and gradually discover relationships that allow them to make predictions, recognize patterns, and support scientific decision-making. For pharmaceutical researchers, this capability is transformative. Machine learning allows scientists to analyze complex biomedical datasets at a scale and depth that would be impossible using manual analysis alone. These models can identify promising drug candidates, predict biological activity, analyze clinical trial outcomes, detect early safety signals, and even assist researchers in designing entirely new therapeutic molecules. Across the pharmaceutical industry, machine learning is rapidly becoming one of the most powerful tools in modern biomedical research. To understand how these systems work, it is helpful to explore the three major approaches used in machine learning: supervised learning, unsupervised learning, and reinforcement learning. Each approach addresses a different type of scientific problem. One of the most widely used approaches in pharmaceutical research is supervised learning. Supervised learning is used when researchers already have datasets that include both input information and known outcomes. In other words, the data contains examples where the correct answers are already available. This type of dataset allows the machine learning model to learn the relationship between the input features and the desired output. Consider a scenario from drug discovery. A research team has collected experimental data describing thousands of chemical compounds that have been tested against a disease-related protein target. For each compound, scientists have recorded molecular characteristics such as molecular weight, chemical structure descriptors, and predicted binding affinity. Alongside these molecular features, the dataset also includes experimental results indicating whether each compound successfully inhibited the target protein. In this dataset, every molecule is labeled as either active or inactive. A supervised learning algorithm can analyze these labeled examples and learn the patterns that distinguish effective drug candidates from ineffective ones. During the training process, the algorithm studies how molecular characteristics relate to biological activity. Over time, the model learns which combinations of features are most strongly associated with successful inhibition of the target protein. Once trained, the model can evaluate entirely new chemical compounds and estimate the likelihood that they will interact successfully with the biological target. This capability dramatically accelerates drug discovery. Instead of experimentally testing millions of compounds, researchers can use machine learning models to prioritize the most promising molecules for laboratory experiments. Supervised learning also plays an important role in clinical research. Clinical trials generate large datasets describing patient characteristics, disease severity, genetic markers, biomarker measurements, and treatment outcomes. Machine learning models can analyze these datasets and learn patterns that predict whether patients are likely to respond to a particular therapy. For example, A predictive model might analyze patient biomarkers and genetic profiles to estimate the probability that an individual will benefit from a specific treatment. Such insights support the development of personalized medicine, where therapies are tailored to the biological characteristics of each patient. While supervised learning relies on labeled datasets, many biomedical datasets do not contain predefined outcomes. In these situations, researchers turn to a different approach known as unsupervised learning. Unsupervised learning analyzes data without labeled results. Instead of predicting outcomes, these algorithms search for hidden patterns within the dataset. One common application of unsupervised learning is patient segmentation. Imagine a clinical trial studying patients with a chronic disease such as diabetes or autoimmune disorders. Although all participants share the same diagnosis, their biological characteristics may vary dramatically. Some patients may respond well to treatment, while others may show limited improvement. Unsupervised learning algorithms analyze complex patient datasets, including biomarker levels, genetic profiles, and clinical measurements, and automatically identify groups of patients with similar biological characteristics. These clusters often reveal previously unrecognized subgroups within the patient population. Discovering these patterns can help researchers understand why certain patients respond differently to therapy and may guide the development of more targeted treatment strategies. The third major approach in machine learning is reinforcement learning. Unlike supervised or unsupervised learning, reinforcement learning is based on a process of trial, feedback, and improvement. In reinforcement learning, the system interacts with an environment and receives feedback in the form of rewards or penalties. By repeatedly experimenting and learning from feedback, the algorithm gradually identifies strategies that lead to the best outcomes. Although reinforcement learning is less commonly applied to traditional clinical data analysis, it has powerful applications in drug design. Drug discovery often involves exploring an enormous space of possible chemical structures. The number of potential molecular combinations is so vast that testing them experimentally would be impractical. Reinforcement learning systems can explore this chemical space more efficiently. The algorithm begins with an initial molecular structure and then makes small modifications to the molecule. After each modification, the system receives feedback based on how well the new molecule performs according to desired objectives, such as improved binding affinity, reduced toxicity, or better pharmacokinetic properties. If the modification improves the molecule's characteristics, the algorithm receives a positive reward. If the modification produces a less effective structure, the system receives a penalty. Through repeated cycles of exploration and feedback, the algorithm gradually learns which molecular modifications produce more promising drug candidates. This approach allows researchers to explore vast chemical landscapes far more efficiently than traditional trial and error experimentation. Across the pharmaceutical ecosystem, machine learning is fundamentally transforming how scientists approach complex biological challenges. Supervised learning helps researchers predict drug activity and patient responses. Unsupervised learning reveals hidden patterns within large biomedical datasets. Reinforcement learning opens new possibilities for exploring innovative molecular designs. These technologies do not replace scientific expertise. Instead, they amplify it. By combining machine learning with experimental research, scientists gain powerful tools for interpreting complex biomedical data, accelerating drug discovery, and improving the effectiveness of clinical treatments. As machine learning continues to advance, its role within pharmaceutical research will only grow stronger. From identifying promising molecules during early discovery to predicting treatment outcomes in clinical trials, intelligent algorithms are becoming an essential component of modern biomedical innovation. Understanding these fundamental machine learning approaches provides the foundation for exploring how these models are built, trained, and evaluated using real biomedical datasets, and how they are reshaping the future of healthcare.